Hello, I am Sittie Fatma Elayna GUIAPAR, and in this video, we will tackle about the placentation, gestation, and parturition. The contents of the video is defining gestation, placenta, and parturition, classification of placenta according to chorionic villi on their surface, classification of placenta based on number of layers, placental regulation exchange of fetus and dam, gestation, and parturition. So let us define first what is gestation, placenta, and parturition. So gestation is the act of carrying or being carried on uterus between conception and birth. Pregnancy and gestation is synonymous, hence gestation means length of pregnancy. This is in contrast to animals that lays egg or what we call oviparous. Implantation is used to mean attachment of the placental membrane in the endometrium of animals, but in humans, it bears itself in the endometrium. Placenta is an organ of metabolic interchange between the conceptus and dam. It is also an endocrine gland that is composed of fetal component derived from chorion and the endometrium from specific zones of metabolic exchange. Parturition is simply defined as giving birth to the young. Conceptus is consist of embryo, which is the initial stage of development of an organism, and embryonic membranes such as the amnion, allantois, yolk sac, and chorion. And these membranes provide protection and means to transport nutrients and waste. Chorion is the fetal contribution to the placenta and the functional unit of the chorionic villus. Chorion villus or chorion villi is a tiny projection of placental tissue that contains the same genetic material as the fetus. This is a significant portion of the placenta that serves primarily to increase surface area by which products from material blood are made available to fetus. Placentas are classified according to distribution of chorionic villi on their surfaces, and therefore classification of placenta. These are diffuse, zonary, discoid, and cotyledonary. Diffuse placentas have uniform distribution chorionic villi that covers the surface of the chorion. An example of this type of placenta is the placenta of sow and mare. The placenta of sow is velvet-like surface with many closely spaced chorionic villi that are distributed all over the surface. Its initial development starts at day 12 and established at day 18 to 20 after ovulation. The placenta of mare is characterized by having many specialized microzones of chorionic villi known as microcotyledons. It also contains unique transitory structure known as endometrial cups that produce the equine chorionic gonadotropin and develop between day 35 and day 60 of pregnancy. By day 60, the endometrial cups are slotted into the uterine lumen and are no longer functional. The figure here shows the diffuse placenta of sow and mare respectively. So the chorionic villi of sow penetrate into the endometrium, forming the fetal-maternal interface, and the vessels from each chorionic villi merge and eventually form a large vessels that enter the umbilical cord. On the other hand, the mare placenta consists of many microcotyledons distributed over the entire surface of the chorion, and these microcotyledons are the site of fetal-maternal exchange. The zonary placentas have a band-like zone of chorionic villi, and this is found in dogs and cats. There are three zones of zonary placenta, namely primary zone or the transfer zone, paraplacenta, the pigmented zone, transparent zone or the non-vascular zone, or allantochorion. The prominent region or the transfer zone, TZ, is where the band forms around the chorion near the middle of conceptus, and transfer of nutrients happens here. The paraplacenta or pigmented zone, PZ, is the second region at either end of the central zone that consists of small hematomas or blood clot and thought to be important in iron transport from the dam to fetus. The non-vascular zone or the allantochorionic zone, the AC, is found at the distal ends of chorion. This zone is involved in absorption of materials directly from the uterine lumen. Discoid placentas from a regionalized disc is characterized by having one or two distinct adjacent discs. These discs contain the chorionic villi that interfere with the endometrium and provide the region for gas, nutrients, and metabolic waste exchange. This type of placenta is found in rodents and primates. The discoid placenta consists a round patch chorionic tissue that forms the fetal-maternal interface. The vessels from the exchange zone, or EZ, merge to form the umbilical vessels that supplies the fetus with blood. The vasculature of the chorion within the disc is immersed in pool of blood where the metabolic exchange happens. Cotyledonary placentas have numerous discrete button-like structures called cotyledons, and this is found in ruminants. Cotyledon is defined as a placental unit of trophoblastic origin consisting of abundant blood vessels and connective tissue. The sheep have 90 to 100 cotyledons, while cattle have 70 to 120. Placentome is the point of the interface. It consists of a fetal cotyledon contributed by the chorion and the maternal cotyledon originating from caruncular regions of the uterus. The chorion initiate attachments to the caruncles of the uterus at day 16 in sheep and day 25 in cattle. The attachment happens or established in day 30 in ewes and day 25 in cows. The placentome forms a concave structure in sheep and goat and convex structure in cows and giraffe. So during gestation, the cotyledons will increase many in fold diameter. This growth provides surface area to support the placental transfer of nutrients from dam and metabolic waste from the fetus. Classification of placenta based on number of layers. So the nomenclature for describing placental intimacy is derived by first describing the tissues of the maternal placenta in the prefix of the word and the tissues of the fetal placenta that constitute the suffix. So for example, the prefix epithelio from mother side and the chorial suffix from the fetal side. So there are three types: the epitheliochorial, endotheliochorial, and hemochorial. Epitheliochorial has 6 layers and the least intimate among many placental types. The endometrial epithelium, or the maternal side, and the epithelium of chorionic villi are intact. This is found in sow and mare. Ruminants have this type of placenta, however, the endometrial epithelium transiently erodes and then regrows, causing intermittent exposure of the maternal capillaries to the chorionic epithelium. And this type of placenta is termed as syndesmochorial. Binucleate giant cells. This is a unique cell type in ruminal placenta. It is quite large and have 2 nuclei that appear about in day 14 in sheep and between day 18 and 20 in cow. It originates from trophoblast cells and thought to be formed throughout the gestation. It also constitutes about 20% of fetal placenta. This figure shows the binucleate giant cells in the placenta of ruminant, in addition to feature the partial erosion of endometrial epithelium. The binucleate giant cells are believed to transfer complex molecules from fetal to maternal placenta. There is an evidence that they secrete placental lactogen, and these cells also secrete pregnancy-specific protein B, or also called pregnancy-associated glycoproteins, that are unique to pregnancy in ruminants. These cells are also an important site of steroidogenesis, secreting the progesterone and estradiol. The second type is the endotheliochorial, which has 5 layers characterized by complete erosion of the endometrial epithelium and underlying interstitium. This type of placenta is more intimate than epitheliochorial placenta because the endometrial epithelium no longer exists. This type of placenta is present in dogs and cats. The third type is the hemochorial. This type of placenta has 3 layers characterized by chorionic epithelium in direct opposition to maternal pools of blood. Thus, nutrients and gases are exchanged directly from maternal blood and must move through this 3 tissue layer only. It is highly intimate and it is found in primates and rodents. Placenta regulates the exchange between the fetus and dam. It involves number of mechanisms found in other tissues, and these are simple diffusion, facilitated diffusion, and active transport. Gas and water pass from high to low concentration by simple diffusion. The glucose and other metabolically important materials such as amino acids are transported by facilitated diffusion utilizing specific carrier protein. And the placenta also contains active transport pumps for sodium as well as calcium. The glucose is major source of energy for fetus and majority is derived from the maternal circulation. The glucose consumption at the near end of gestation is exceptionally high and can lead to metabolic drain of glucose away from the dam. Some materials cannot be transported across the placenta except the immunoglobulin. Immunoglobulin transported from maternal to fetal side in hemochorial or endothelial placenta. However, fetus synthesizes the majority of its own protein from amino acids contributed by the dam. Nutritionally based lipids do not cross the placenta. Instead, they synthesize a new lipid material for the fetus. Large peptide hormones such as thyroid-stimulating hormone, adrenal-cortical-stimulating hormone, growth hormone, insulin, and glucagon do not cross the placenta. Smaller molecular weight hormones such as steroids, thyroid hormone, and the catecholamine cross the placenta with relative ease. Vitamins and minerals are transferred to the fetus at a variable rate, and fat water-soluble do not cross placenta with ease unlike water-soluble vitamins. Nutrients are also transferred by phagocytosis and pinocytosis. Areolae from chorion form over the openings of the uterine glands are thought to absorb secretion from these glands. Toxic substances, potentially pathogenic materials, and viruses can easily cross the placental barrier. Some substances may be highly teratogenic. It means it induce the abnormal embryonic development and can cause serious birth defect. Wide range of microorganisms can contaminate the fetus.